Hello. Today we're going to keep talking about the immune system, but the focus is on humoral immunity. Have you seen people, have you seen advertisements where people are asking anyone who's had COVID to donate their plasma? And do we know why? Like, what's in their plasma that people want? I'll let you watch this video
I'm Dr.
on your own time if you would like.
I'm Dr. Steve Hahn, Commissioner of
Maybe.
Food and Drugs.
And then some people mentioned this when we did that little immunity research project, that there's something about antivenom where you can take venom from a snake and then you actually infect an animal with it. So you poison the horse in this case, and then you take the horse's blood and you take the antibodies out of its blood and put it into someone who's been bitten by a snake. And a similar thing going on here with breastfeeding. One of the benefits of breastfeeding is that the mother is able to transfer antibodies to her babies. So these are examples of the humoral immune system. This is a nice animation of that if you want to watch it. I'm going to keep going. If you click watch this video on YouTube, then you should be able to. All right, humoral immune response. It's all about B cells, and the B cells secrete antibodies. So this is a B cell, and all of these little Y-shaped things are antibodies. B cells are able to target extracellular pathogens. So, for example, that could be like a bacteria or a parasite that's invaded the body, or maybe like a virus if it's floating in our fluid. They would not attack like infected body cells, like cancerous cells or virus infected cells. That's going to be cellular immunity. So humoral makes antibodies and acts on non-cellular pathogens. Let's take a look at this diagram. So this kind of represents humoral immunity. So all of these three cells right here are B cells, and notice that they have antibodies sticking out of them, and they're three different colors to represent like three different shapes. Remember that that shape was determined before they ever encounter their antigen, like it was determined by the genes. But then one day, this B cell in the middle happens to encounter an antigen that it's able to bind with. So this antibody has the complementary antigenic determinants to be able to bind with this antigen. This act of binding activates this B cell. This is still naive because it hasn't been activated, but this one found its antigen, it's been activated, and it immediately starts dividing. Most of the cells divide and form effector cells, like the cells that actually have an effect. And with B cells, those cells are called plasma cells, and their function is to secrete antibodies, and then the antibodies are going to circulate through the blood and the lymph. Look at these cells. That's the nucleus. This organelle is to represent the ER, the endoplasmic reticulum. Plasma cells have lots of ER because they have to make lots of proteins, aka antibodies. Antibodies are proteins. They can secrete antibodies at a rate of 2,000 molecules per second. They live for around 4 to 5 days, and like their entire lifetime, all 5 days, they're producing 2,000 molecules per second. So I'm amazed they can stay alive that long. Notice that not all the cells became plasma cells, most of them did, but a few differentiate into memory cells. These are going to be able to remember the antigen and launch an even faster attack the next time it happens. So these do not produce antibodies. They're just going to go and hide in the lymph node and wait to see if we encounter the antigen. This diagram represents what's called the primary immune response, and that means what happens the very first time you encounter an antigen. It's a little bit different the second time you encounter the same antigen. So this diagram is showing the secondary response. So here we have a memory B cell who's already been activated. So basically, it's going to immediately start generating plasma cells. It's making more plasma cells than last time, and it's making them faster. And because it's making more plasma cells, there are even more antibodies available. And the antibodies are going to remain in our blood for a longer amount of time. And we always have memory B cells so that they can remember it again. So the secondary immune response, it could be the second or the third or the fourth or the fifth or the 20th or the 40th or the 100th time. It will be like this exact response. So only the literal first encounter is slow. Let's take a look at this graph and use it to compare the primary and secondary immune responses. So this blue line represents the response to antigen A. Here's the primary response to antigen A. Here's the secondary response to antigen A. And by the way, this is time in days, and this represents the concentration of antibodies. And this purple one is a response to antigen B. Over here on day 0, we had our first exposure to the A antigen, and then it looks like it took about 6 days for the body to start producing antibodies. That's pretty normal. There's normally a lag of 3 to 6 days because the B cells do need time to differentiate and produce all the plasma cells that they can then produce the antibodies. Also, notice our concentration is right about 10. So you could think of this as like 10 antibodies per liter of blood. So better than nothing. And then the peak was reached about 10 days after the initial exposure, and then it declines. So hopefully 10 days was enough time to fight off the invader. If not, we might have a little problem. Then right here on day 28, this represents that we got exposed to the same pathogen again. And notice there's almost no lag time. Like almost immediately, the body starts producing antibodies, and their concentration is way higher. Like we're up here close to 10,000 antibodies per liter of blood. And notice this, they're staying high for weeks, and then they're starting to decline as opposed to only staying high for 10 days. So pretty cool. Yeah. What we've been talking about so far is described as active humoral immunity. That means that our B cells literally encountered the antigens and started producing antibodies against them. So, active humoral immunity means the B cells encountered the antigen and they started producing antibodies. We can acquire this 2 different ways. The natural way is what we've just been describing, where an antigen like a bacteria enters your body and then the B cells are activated and they start producing. How else could we get it? I hope that you're saying vaccine, because that is the answer. The artificial way that we can counter it is through a vaccine, where like a dead or weakened form of a virus is inserted into the body, but that virus has the same antigenic determinants, and it is able to activate our B cells and then create those memory cells. And because the virus is either dead or weak, we typically don't have as many adverse symptoms as if we were actually sick with the virus. A few adverse symptoms are totally normal and probably a lot better than getting the virus. We can also get passive humoral immunity, and this would be like taking antibodies and injecting them into your body. So you're not actually making them. The downside is that your B cells are not activated, so we don't have any memory. And also, this like borrowed immunity is going to end when the antibodies degrade. So this would be like if someone is going to die, like a disease kills you so fast that you're going to die before you can make your own B cells, that's when we would transfer them to someone. This can also happen naturally, like when a woman is pregnant, antibodies cross over the placenta to the fetus. And then also if a woman breastfeeds, antibodies in the breast milk are able to transfer to the infant. And then some of those activities we were talking about at the start of this, that represents artificial passive immunity. So exogenous means from outside our own body. So that would be like the antivenom, where we take the antibodies from a horse who was infected and give it to like a person who was bitten by a snake. Or we take antibodies from someone who's had COVID and we give it to someone who's infected to help being able to fight it off. The effect is short-lived, but again, like you live instead of die. Here's a summary of that in concept map form. Getting vaccinated is really just teaching your antibodies to hug viruses. And antibodies love hugs. Aww. Ooh, and here's a little review game. Someone comes into the ER with a rattlesnake bite. The doctor gives her rattlesnake antivenom made from antibodies. So would that be passive or active? He's giving her antibodies, so passive. And is that natural or artificial? That's artificial. So we have passive, artificial. Number 2, a person receives an injection of killed poliovirus, which causes them to generate memory T cells and B cells. They're generating their own cells, so it's active, and this is artificial. Vaccine, active, artificial. A baby receives IgA antibodies from its mom. This is passive and natural. Passive because we're just giving them the antibodies, and natural because it was coming across sounds like the placenta or the breast milk. 3-year-old gets the chicken pox, activating his immune system and causing the differentiation of memory T cells and B cells specific to the chickenpox virus. That's effective because we're making the cells and it's the natural way of doing it. All right, true or false, antibodies can destroy antigens. I hope that you're saying false because they actually can't, which kind of makes them seem weak. But they can do other things. So they can't literally destroy them, but they can bind to them and like mark them to be destroyed by other things. And it actually is more important than it sounds. So we're going to talk about how antibodies are able to do this. Like if they can't destroy the antigen, what do they do instead? So here's our Y-shaped antibodies, and they're going to bind to an antigen, and then they can do 4 different things. They can do neutralization, they can do agglutination, they can do precipitation, and they can do complement activation. So notice in all 4 of these, we have an antibody binding to an antigen and forming this immune complex. Now let's talk about all 4 in a little bit more detail. So focus on neutralization. This is a really simple antibody trick, but it's one of the most important defensive mechanisms that we have. So basically, an antibody is going to bind to a specific spot on a virus or on a bacterial exotoxin. That would be like a toxic chemical secreted by bacteria. And since the antibody has bound to the virus or the toxin, that means that it can't bind to the body tissue. And then eventually the cell is going to be destroyed by phagocytosis. So here we can see antibodies where let's just say they're binding to a virus down there, which is preventing it from being able to infect any of our cells. And then also, this is like the complement proteins, opsonization. A phagocyte can see this and they're like, oh, I should eat that. It looks good. Yeah, and actually opsonization literally translates to make tasty. So it's literally what the phagocytes are thinking when they look at it. Okay, agglutination. Antibodies can bind the same determinant on 2 different antigens at the same time. So the antibody has a Y shape, and so like the upper arms of the Y, those are 2 binding sites. And because it can bind to 2 things at the same time, it's able to form clumps. So we can see that multiple antibodies have bound to multiple antigens, and then they're all clumped together. And this large clump is easy for the phagocytes to be able to find. The clump is called agglutination, and you might remember it because this is what happens when you mismatch red blood cells. So we did that blood typing lab, and we added antibodies, and we looked for clumping. Precipitation. Precipitation is just like agglutination, except that the antibodies are binding to molecules instead of to like red blood cells. But we see the same thing happening down here where antibodies are binding to multiple antigens and they're forming a big clump. The clump precipitates and it makes it easier for the phagocyte to be able to find. And then the last one, complement fixation and activation. So this is how our body typically tackles cellular antigens like a bacterial, or if you got a blood transfusion with the wrong blood type. When antibodies are bound close together on the same antigen, the complement binding sites on their stem regions are aligned. So here is, let's say, a bacteria cell, and then here are two antibodies that have bound to it, and they've bound side by side, and this, like the stick-like portion of the Y is right next to the other one. When they're bound together close like that, it actually triggers complement proteins. So them binding together is the signal for complement proteins to be activated. And once the complement proteins are activated, they form MAC, MAC. That's the membrane attack complex. It's like a pore into this cell where they are able to secrete chemicals, and then it causes the cell to lyse or to die. When the antibodies call the complement proteins, that enhances the whole inflammatory response and it supports opsonization. So it supports the complement proteins and the antibodies binding to other things. So here's that picture again from the beginning, so we can kind of see how it all goes together. So neutralization, agglutination, and precipitation, all resulting in phagocytosis and helping to increase inflammation. So the complement proteins causing histamines, causing mast cells to release histamines, which are helping to guide macrophages to the correct area. And then over here we can see our MAC causing cell lysis. The cell is dying. That's good. That's bacteria. A pretty way to remember how antibodies work is to remember they have a plan of action: precipitation, lysis by complement, agglutination, and neutralization. That is from our textbook, but I think it's a really helpful way to remember what antibodies can do. And this is a cool video that talks about how we're using antibodies to be able to fight off COVID. And please let me know what questions you have.